apheresis. So I'm going to discuss with you, along with Dr. Eric Verdin, we'll bring him back here in a few minutes, a project that took about 2 years. I think I talked a little bit about it 2 years ago here at this conference. And it was a collaborative project between my company, Circulate, my older company, Global Apheresis, and the Buck Institute. So before we get to the study itself, I would like to tell you a little bit about the history of how we got here. In 2005, Irene and Michael Comboy, along with their colleagues from the Tom Randall Laboratory in Stanford, did a very interesting and exciting experiment. And this experiment is called parabiosis, and for those of you who don't know what it is combining the circulation of two genetically identical mice, one older and one younger. After a short period of time, they observed both morphologic as well as functional changes in both mice. The older mouse became younger, very exciting, and the young mouse became older. Very few people paid attention to the latter. Everybody was excited about the old mouse becoming younger. So the fountain of life, the fountain of youth, was into the new blood. This led to very kind of interesting experiments. Certainly not to my taste, but people ended up in front of blood banks and looking for young plasma, and this thing went on and on. It's still going on in some places. The FDA had to be involved because there were some serious side effects, including death. So it's not advisable, but it's fully understandable after this exciting experiment. So I think that our friends that tried to do that have forgotten that we are not mice. And that's very important because there is no single animal experiment that directly translates into humans. We learn from animal experiments, but we have to modify what we learn from them to apply it to humans. And that's what we try to do in this case. In 2013, I published a hypothesis which actually is attempting to simulate parabiosis in humans using therapeutic plasma exchange or plasmapheresis. These are interchangeable, so you can say TPE or therapeutic plasma exchange or plasmapheresis means the same thing. I'm going to play a little video to tell you what apheresis is all about.
Therapeutic plasma exchange, or TPE, refers to a specialized medical procedure in which blood separation technology is used to remove abnormal plasma constituents. Whole blood is removed from the patient into a blood cell separator, which utilizes centrifugal forces to separate whole blood into its individual components: plasma, platelets, white blood cells, and red blood cells. Plasma can be removed and replaced with physiologic fluids to treat relatively uncommon but usually life-threatening diseases.
So this is how it looks in real life. You know, people are in a bed. They have two IVs put in their arms. It's a machine. If you're lucky enough, you're gonna get a little service dog in your lap. So obviously, as you saw, we can remove the plasma of an older individual, of any individual for that matter. And in older individuals, it is important to remove as much plasma as you can, because with time, the plasma accumulates a variety of pro-inflammatory and progenitoric factors. And this is well studied and documented. So we want to clean the plasma, and we remove 2 to 3 liters of plasma during each procedure. So the question came, what do we replace it for? We have to replace it with something because we remove a large volume, and we have to substitute for something, otherwise we're going to have issues. Ideally, you want to replace with young plasma. However, young plasma is associated with very serious side effects. Some of the side effects are common and not that bad, but eventually there are serious side effects, as I mentioned, including death. So what we decided to do is, instead of using young plasma, we use 5% albumin. Albumin is made of plasma, and the plasma is collected in donor centers throughout the world, and the average age of the donors is 25 years old, which is very young. So chances are, if we really think that there is something in young plasma, it probably will be in the albumin, which is the largest portion of the plasma anyway. So albumin for many years has been known to be very powerful antioxidant. It's an anti-inflammatory agent, and it's immunomodulatory. So using albumin made a lot of sense to me. We did a few patients to begin with, and I presented the results of our initial studies here 2 years ago. And we were encouraged with what we saw. However, until it was proven, it had to be proven by the gold standard of science, and that is this was published a couple of years ago. We had to prove it through the gold standard of science, and that is double-blind, placebo, sham-pheresis controlled clinical trial of patients. And we chose patients over 50. The average age was 72, and the oldest person we treated was close to 90 years old. Now, the sham-pheresis is a very complex procedure by itself. I don't have the time to get you into this. My nurses were very brave and very technically savvy to perform the procedure. And my chief nurse, Cello, presented an abstract at the American Society for Apheresis last month discussing the difficulties and intricacies of sham-pheresis. Just to show you here that the patients are divided from the machine with a curtain, and their venous lines are covered as well, so they don't know what is going on in and out of their body. We enrolled 40 patients in this clinical trial, and we divided them in 4 groups, 10 patients in each group. The first group, they underwent 2 plasmapheresis treatments in 1 week, and this was repeated for 3 months, so a total of 6 procedures. In this particular group, in addition to albumin, we used small amounts of intravenous immunoglobulin, or IVIG. Group B was exactly the same, but without the IVIG. Group C was the sham-pheresis. They didn't get real pheresis, but sham. And the last group underwent 1 monthly procedure for 6 months. This was our original protocol when we first started doing this. And we followed a variety of parameters. Of course, we were very interested in the safety. We followed a variety of biomarkers that Dr. Verdin is going to discuss in a second here. And we had a few macro parameters that I'm going to show you. We have a single mild adverse reaction, a mild allergic reaction to albumin. Albumin, from time to time, is difficult to to get. During the pandemic, it was a problem to access it, and we had to change the company that makes the albumin, and the new albumin usually causes some problems, and in this case, it was a mild allergic reaction. We measured strength, and we measured balance, and we measured something called up and go, and that's how it looks like, how fast one moves from point A to point B, and then we measure it with a chronometer. And these are the results from the hand grip. As you can see here, all treatment groups improved and improved statistically with proper p-value. And this was not the case in the sham pheresis. The up and go, significant decrease in the timing of the up and go in all three groups, and again, no significant change in the sham pheresis group. In the balance test, we have some changes, but it was not as spectacular as the other measurements. And in group D, we didn't see any significant change, actually. We use something called SF12, which is a questionnaire, which is then submitted to artificial intelligence and measures overall physical score and mental score. So in this particular test, the physical score was not significantly improved, although improved, not significantly improved in any of the groups, except for the last D group. Interestingly enough, there was a dramatic increase in the mental score, which obviously is very important, especially in this group of patients. At this point, I will invite Dr. Eric Verdin, who is going to discuss with us the biomarkers that were measured by his colleagues in the Buck Institute.
Thank you. Is my mic on? Yeah, okay. Thank you, Dobri. First, I want to thank Dobri for the opportunity to collaborate with him and his team on doing this. I think one of my pet peeves of our field is the fact that sometimes the selling goes ahead of the science. And I think I've argued that as a field, we will only gain the credibility that we crave by doing rigorous clinical trials and really demonstrating the efficacy and the safety of what we're doing. And so, doing a sham pheresis is hard, and in some ways the true test of really what works. So when Dobri approached us, the idea was to really deploy everything that we could possibly deploy in terms of high-level resolution of biomarkers and try to determine whether there was a signature associated with this plasma pheresis. And I should say that I don't want to raise the anticipation that about what I'm about to tell you, because the results of the trial was completed pretty recently, and we are confronted with a unique problem, is that we have too much data. And so we are sifting through it. I want to acknowledge the work of David Furman and Herb Kassler, Prasanna, and Birgit Schilling and our team at the Buck who are still working actively at deciphering these data. Now we're dealing with tens of thousands of data points for each sample. So this is a problem that is a good problem to have, but in some way we've reached the level of interpretability of these results. But I thought there were some preliminary results that I want to show you that were significant enough to warrant sharing them with you. And obviously we're in the process of preparing a publication with Dobri, which I think will explain all of the results. So what we did was deploy all of the omics, metylomics, these are the clocks, and I'll be speaking especially about this, proteomics on plasma, lipidomics, metabolomics, glycomics, and cytomics. Cytomics is the high-level deep immune analysis that I've talked about. And the questions that we were trying to address were several. One, what can we see in terms of response? Do we see a biological response in terms, in addition to the phenotypes that are clearly improve that Dobri just shared with you. The other questions that we are trying to address with this type of technology is to decipher if we have a molecular signature of response, how long does this response persist? That's one key question. How frequently should we do these types of treatments? The second one is, can we actually identify early in an experiment before we treat the patients, what are the which subjects will actually respond in a positive way to this? And again, we are dealing with small groups of 10 humans for any clinical trials. It's actually right at the limit of detection. But despite this, I want to share with you a few of the positive response that we've seen. So the bottom line is that the system types of analysis revealed baseline features that drive biological age rejuvenation in response to plasmapheresis. The key slide that I want to share with you is one in which we applied all of the clocks. And I want to say a few words about what the clocks mean today, because there's proliferation of these clocks, and which is again compounded in the public domain by the fact that many companies are starting to commercialize this. And I get a call a week from someone telling me I've had my clocks measured and they're all over the place. I'm personally, I did all my clocks as well. I'm between 25 and 67. Of course, I choose 25, but it's important really for us to discourage the public from using these tools, frankly, because they cannot interpret them. Doesn't mean that a longevity clinic with people who are properly trained can actually not use them, but they are experimental research tool at this point. Despite this, so what we did is to take what I would call an agnostic approach. We didn't really take a favorite clock. We just used all of them. This is work done in collaboration with True Diagnostic. And what I want to point to you is the bar here. So we have, again, the 4 different treatments. This is group A, B, C, and D. The sham actually is switched here. It's the last one here. One thing that I want to point is that the sham itself is not entirely benign. I think this is an important thing to consider in a way that it results in a mild level of age acceleration, which we observed. So this is something, obviously, plasmapheresis involves the subtracting, a significant amount of blood and restore. And so there's a lot of volume. It is probably perceived to some degree as a stress by the body. And that's something to consider as we are trying to discuss with Dobri, how frequently should we do this and how many times. But so that there's a clear signature of some degree of stress that we can even measure in the clocks. But despite this, when you do the true pheresis, and this is the first column here, especially with this group A, you can see a clear negative age acceleration. Now, the field talks about age acceleration, so a negative age acceleration is actually age reversal to some degree. And I'm showing you, this is with as many as 2 visits. You can see that this is more summarized on the bottom right panel, where each of the treatment shows actually a significant age deceleration of 2 years, and the sham actually shows a mild 0.5-year acceleration. So if you look at the whole net results is still quite significant for a short-term treatment. And obviously this is going to be the first step in what we think will be an iterative set of clinical trials to try to define, okay, what can we do now? It seems that there is still a lot of room for us to improve how this treatment works. The next step, what we did, is to deploy all of the omics that I've told you, and they're shown on the left. Each circle represents many of the things that we've measured and identified to correlate with the results of the intervention, and try to determine which of these changes that we detected early are predictive of a positive response. Again, as I mentioned, a trial with 10 patients is hard to do. It's a rate-limiting step. Obviously, we'll do more in the future. But despite this, we were able to identify a whole series of glycomics, metabolomics, and they're shown here individually. I won't go through them, but we now have a set of markers that allow us to determine whether a subject is going to be responding or not. And obviously, this will be critical in the future. One thing that I want to share with you also is some results that we've conducted on the SASP. So the SASP, as I mentioned earlier, is the set of proteins that are secreted by senescent cell protein. And Birgit Schilling, my colleague at the back, along with Judy Campisi, has been a pioneer at defining the composition of the SASP. It's actually a lot more complex than we thought. The experiments that were done in the past was to generate the SASP both as soluble protein and as exosomes from different cell types in an in vitro setting, to do the proteomics, identify all of the proteins, try to determine a common set of proteins that were secreted by these cells, and then intersect it with proteins that we know are circulating in plasma. So this generated, that was a verification. And the last step was to take untreated or young versus treated or old individuals and trying to validate the predictive value of each of these markers in terms of disease association and so on. And so this led to what we call the SASP Atlas, which is publicly available. It was published in PLOS Biology in 2020, and any of you who want to use this, feel free to access it. I think it's an important tool along with the Sen Mayo that I described earlier. Now, when we do this and we look at the relative abundance of each of these SASP proteins, so this is a small subset of the SASP protein. Each bar on this graph on the y-axis is a different SASP protein. And what I'm showing you here is their relative abundance after TPE treatment. And you can see that we don't see a global suppression, which indicate, and this is consistent with what we've seen in other experimental processes. The SASP itself is cell-dependent, so it might be that we're targeting some really unique cells, but we clearly see a suppression. You see the bottom left corner has a whole series of proteins that are, many of them heat shock proteins or glycolytic enzymes that appear to be quite significantly downregulated as a response to the same treatment, the group A, in which we saw the decrease in biological age. So so we're obviously spending a lot of work now trying to use all of these omics and try to see how they interact with each other to try to define the biology of what this intervention actually means. There's still a black box, frankly. The idea that these treatments do work is remarkable, but the question is, what are we really doing? And I think this is what the system biology analysis will provide us in the future. I want to summarize briefly. So we've identified the best treatment modality, which is this TPE biweekly plus intravenous immunoglobulin treatment, decreases acceleration. We identified a most sensitive measure to TPE, so the system age clock, which were recently described, actually decreased the most. We constructed a TPE response scoring system, and the multiple omic feature track with improvement in TPE response score. And so baseline testing, we hope in the future, can help us to stratify patients for improved treatment efficacy. And I want to stop here and actually have Dobri come back and finish.
Thank you, Eric. You can stay, maybe we can take questions together. This is just a summary slide that just about every aspect of aging is affected by plasmapheresis. Unlike any drug that you can submit, this is a process that is very complex, multidimensional, and affects every aspect of senescence. And basically, the old plasma, old milieu where the cells grow is becoming new. I would like to thank the sponsor of the study, Goslar Ventures. I would like to thank all the people at the Buck Institute who helped us a lot. As you saw, they performed incredible tests and still doing them. Steve Horvath's lab and Bobby Brook, who is here. True Diagnostics, Ryan Smith and his team that did some phenomenal work with the clocks. And this is my team. You can say here. I'm very proud of my team. And as I mentioned, there is a little dog, and some people say that he's responsible for some of the positive results we see. I don't know about that, but I'll take it. Thank you very much.